continuation of my lecture on protein molecules. Let me talk on the applications of protein molecules. The first application is protein molecules as structural and mechanical support. Protein molecules as structural and mechanical support. What is meant by structural and mechanical support? It is a structural and mechanical support to cell as well as tissue matrix. Let me recall the tissue, definition of tissue in brief. The cells are embedded in the tissue matrix. The tissue of an organ, where the tissue matrix are tissue mass in which the cells are deeply buried and fixed firmly in that matter. That is called as tissue or cells. Cells are microscopic, of course. In this we have different types of materials. In this tissue itself, we are calling as two different types of materials. One is cellular material and tissue material. The material inside is called as intracellular matrix and the material outside is called as extracellular matrix. Like different molecules, of course. Now today we shall concentrate on the intracellular matrix. We shall concentrate more on the intracellular matrix. The cells are there, cellular material, all the cells of the same material. Let us take the individual cell. What is individual cell? The cell, you see that is the compartmentalization of cell. Cell membrane and organelles, cell organelles, nucleus, endoplasmic reticulum, rough and smooth, as well as there are Golgi bodies, mitochondrial bodies, lysosomal bodies, and here lysosomes, cytoplasmic. There are lysosomes, cluster of lysosomes in the cytoplasm as well as on the endoplasmic reticulum, membrane. Yes. And also some structure is there, that is thread-like structure. It is called as cytoskeleton structure. That is what I am talking about today. There is more stress on the cytoskeleton. Now I want to talk more. What is that? This is regarding the cell, the cells and the organelles. Here, all this, just a brief about this. All these organelles, look at this, whether is endoplasmic reticulum, mitochondrial body, Golgi bodies, they have their different shapes and what is their sizes. They have ribosomes, lysosomal bodies. All of them are membrane bound, membrane like sacs, membrane. Just like membrane is cell membrane. What is cell membrane? Cell membrane is a bilipid in which protein molecules are present. Some protein molecules, it is a combination of bilipid. What is that? Lipid bilayer lipid molecules and protein molecules. Protein molecules for the structural support. It also contains, these membranes also contain same kind of membrane. Lipid bilayer with protein molecules. And every organ is an organelle here now. They have their own enzymes. They have a set of enzymes inside and protein molecules for their structural support. Every organelle, whether it is mitochondria, Golgi bodies, endoplasmic reticulum or lysosomes, these bodies. Every substance, what is that? Every organelle have their own enzymes inside and also some protein molecules for their structural support. These enzymes, the variety of enzymes, the number of enzymes are there. These are the enzymes that can carry out the specific biological reactions in these organelles. So every organelles have their own specific biological reactions. They are doing it. They have their specific reactions. So these enzymes, different enzymes, all enzymes are protein molecules. That is a different functional. That is a different application of protein molecules. And here This is one I am talking about the lysosomal body, a sap containing enzymes. These enzymes are different. Lysosomal bodies. What is that? About 50 enzymes, 50 different enzymes are there, even more than that. What is the purpose of these enzymes? What these enzymes will do? These enzymes can break, what is that? Break the macromolecules. All the macromolecules, what is that? That is made up of macromolecules. All these organelles are made up of macromolecules. Every item in this made up of macromolecules, set of protein molecules and different molecules. And these enzymes can break those macromolecules into simple molecules, break slices into simpler substances, simple molecules. That is the function of these enzymes. The purpose of these enzymes, just what is that? Remove any damaged organelle in the cell. If an organelle is damaged accidentally or by infection, it is possible. If that organ should be removed, it is damaged beyond its limitation. What is that? That organ should be removed. It is removed by the action of these enzymes. These enzymes will come out and break into pieces, slices into pieces, simple molecules. And the molecules, thus what is released, may reuse as a nutrient and some as waste. So among the molecules, what they are releasing by the action of these enzymes, some have what is a useful nutrient can further use by the cell or as a waste product. Will be removed as a waste from the cell. That is possibility. That possibility is there. These enzymes not only break the macromolecules of these, sometimes these molecules, what is that, break all the organelles when the cell is damaged, cell itself is damaged. The cell is damaged, it should be removed in the system. In that case also, these enzymes will actually, what is that, this pieces, slice all of them into, the entire cell into pieces, molecules. And these molecule enzymes not only act on these macromolecules, they can act on the macromolecules which are made, which are macromolecules of the invading bodies, maybe pathogens or some other bodies, sometimes is invading the cell. Those macromolecules also it can break into pieces. That is a kind of enzymes. It is storing up. This is a bilayer, lipid bilayer with bag like this, like all the enzymes kept reserved. If any such events arises, they quickly come out and act on immediately. That is the one requirement to save the cell in the case of danger. That is the purpose of this. This is about just a brief about the organelles, functions of organelles. Now come back to the point. If the cell, what is that, is a spherical shape, of course, approximately a spherical shape, inside a three-dimensional space around the nucleus. Here, total space is there. All these contents are there. Suppose if you fill this space along with contents with aqueous solution, there is cytoplasm. What is cytoplasm? The aqueous solution containing all the nutrients required for biological activity. If you simply fill with aqueous solution, homogeneous aqueous solution, what will happen? Will the cell remain as if or retain the shape? Highly impossible, because the contents of the cell, contents means these organelles and aqueous solution exert pressure, hydrostatic pressure, mechanical pressure with which the cell cannot withstand, collapse. If that is the case, what is the requirement? That is what we are talking about. Requirement is a support, mechanical support. Some mechanical material of tensile strength, some required tensile strength required for the support of the cell to retain the shape and size and to keep the function always going on. That is called as structural and mechanical support. The material is required. What is the material? Material is cytoskeleton. What is cytoskeleton? Cytoskeleton, just like lines I showed that in all three dimensions. It is a three-dimensional mesh-like structure. A three-dimensional frame-like structure, scaffold, structural material, a material for mechanical support that is required, most important. That is a three-dimensional structure, and that mechanical support, that is that mesh or three-dimensional mesh, which have certain mechanical, what is that, tensile strength which can withstand. That material or three-dimensional structure can withstand whatever the amount of pressure or mechanical force that is built by the load of the material. That is the content of the cellular content of the cell. It can withstand. Then the cell can retain the shape and size. That is the kind of what is that mesh is required. That is a three-dimensional mesh or scaffold is just like a flat form for the cell. That three-dimensional structure is just exactly same as a flat form or a stage for a drama, just like a drama. Here, if the stage is constructed, if a proper stage is constructed, every actor can come and act effectively. The effectivity of the act depends on the stage which can give support for their action. That is why it is same as that three-dimensional structure, or what is our skeletal structure I am talking about, mesh-like structure. It is the one which can give support for the activity for all these organelles, this biological act of all these organelles. It is a flat form, scaffold for the action of all these organelles. It is called skeleton, cytoskeleton. What is the cytoskeleton material? It is made of, just like a stage or made of somebody is a solid materials, rods like this. So it is made of, say, filament or thread-like material, a microfilament. Width is 8 nanometer. And a tube-like structure or rod-like structure, microtubes, 25 nanometer width. And one more intermediate filament, it is just appear like what is that, a rope-like structure, 10 nanometer width. These are the structural material. With this structural material, this is built, cytoskeleton. The mesh-like property is built. These are made of protein molecules. All these are protein molecules, variety of protein molecules, lakhs of different protein molecules. Let us look into that, what type of molecules are involved in this construction of, in this making of these structural materials. And one more point I want to stress here. This mesh or scaffold not only give a mechanical strength to the cell, very important, it gives what is some kind of ideal medium for the biological reaction. What is the ideal medium? It is the mesh on mixing with aqueous solution or cytoplasm. What is cytoplasm? Cytoplasm is aqueous solution of all the nutrients. It is aqueous solution, liquid layer. That a mesh becomes a gel because protein molecules. So water soluble, water is partially soluble, completely soluble. What is that? Water wet material. So what is that? Becomes a gel like. What is gel? Gel in chemistry, as a subject is there. Chemistry, gel means one phase is disposed in other phase. You have different phases: solid phase, liquid phase, gas phase. One phase is disposed in other phase. One phase of certain size disposed in other phase. Gel is liquid is disposed in solid. Solid is cytoskeleton. Liquid is dispersed. It is called gel, and fluid is called a gel-like fluid. Now, the cytoplasm is not simply aqueous medium; is a jelly-like fluid. That is the one is requirement for the normal biological reactions is taking place all over. Very important. It is an ideal medium, gel-like medium is obtained. It's because of cytoskeleton not only as a mechanical support act as a jelly material for proper biological reactions in the cell. That is called as jelly material. Mechanical structure. What is this cytoskeleton? Let us go in detail. What is a microfilament? Microfilament is made out of globular protein. In the previous class, I stressed upon the protein molecule after it is being synthesized. Every protein molecule assumes its own geometrical shape. Geometrical shape in these Golgi bodies. It assumes its own geometrical shape. It may be in a globular shape or fibrous in the last class. A globular shape or fibrous shape depends on what kind of amino acids, which amino acids are enriched in the molecule. Decides either a globular protein or fibrous. Globulin is a globe-like, a spherical-like, and fibrous protein, a rod-like or spread-like proteins. So there are only two types of what is that molecular geometry we are expecting among the protein molecules. One is globular proteins and fibrous protein. And this geometrical transformation is occur always in Golgi bodies. After it is being synthesized, they enter into Golgi bodies where it is under a modification. What modification? There is substitution. Any substitution is required, is substituted and make the molecule into a certain specific shape, biological, which is required for biological functions. That is the Golgi bodies. Now talking about this microfilament. In this microfilament, it is a globular protein, not fibrous protein. Important. What happens here? Globular proteins, how it acts as a microfilament. Very interesting. Globular protein is what is the globular protein molecule? Now it is called as G. Here this is called as G-actin molecule. A molecule like this. It contains what amino acid? How many amino acids? 375 amino acids. A globular macromolecule consisting of 375 amino acids. And after its globular shape, it has some calcium ion and ATP ion on the surface. These two are required for the polymerization. Here it is called as monomer. Very interesting that this globular protein undergo polymerization. This is an interesting question. In chemistry, usually what happens? Simple molecules undergo polymerization to grow into big molecules. Where polymer molecule itself undergo polymerization into a big polymer. That is why it is happening here. That is acts as a monomer. It's not a simple molecule, it's a macromolecule. Polymerizes macromolecule. The same molecule polymerizes. This polymerization, linear polymerization, forming a longer thread-like stiff structure, like molecule is got, it's a polymer molecule. It is finally, and after that complete synthesis, it occupies what is that? Assumes helical structure like this. Two such helical structure combined like this, twist around each other, wind around each other, forming a filamentous structure. There is a filamentous structure of width varying between 7 to 10 nanometer. It is a thin filament, it is called as this biochemistry. This is one kind of structure. This is obtained because all these molecules are synthesized here only, and this all geometrical result synthesis of this also in the cellular for this purpose. The cell itself make these molecules for this structure. All this synthesized in the cell itself. This globular protein and polymerization finally into filamentous like this, twisting like this. This is one structure. Another structure is microtubule, tubular structure. What is this tubular structure? In this again, the protein molecule is a globular protein, not fibrous. There are two globular proteins are involved. One is called as alpha. Alpha, it is called as tubulin. It is called beta tubulin. Globular protein. Each globular protein contains 427 amino acids. But differ is amino acid sequence. The sequence of amino acids is different, but all contain same amino acid. Same number, same amino acid. Globular. Alpha tubulin, beta tubulin. This is after synthesis, then what is that? They chemically combine. They can again, these macromolecules chemically combine, forming a dimer called as tubulin dimer. Alpha beta dimer. Alpha beta dimer. It is called as monomer. Now monomer. Here monomer is one globular protein. Here monomer is dimer. Alpha beta dimer. Each containing 427 amino acids. That is one unit. Another unit, dimer. So beta is shaded, alpha is not shaded. Just to specify that. Here unit polymerizes in a linear fashion, forming what is that? A filamentous like structure is called as photofilament. Polymerization like this. It is called it is lakhs or thousands like this polymerized into a photofilament. After the photofilament will not remain as if. Undergo further polymerization, further interaction, further association. These photofilaments undergo further association, cluster of photofilaments forming a shape like this. It is an allocore, allo shape like this or tubular shape like this. This allocore or tubular shape consists of 13 such photofilaments. That is one photofilament polymerized, one photofilament, there is one more, all around 13 photofilaments. That is just like the upper surface here, sideways. Lumen, inside is allo, space, empty space here. Finally, what tubular shape? It is the width. Finally, it is turns out to be 25 nanometer, bigger size, and the length is different, varying from 200 nanometer to 25 micrometer. A rod-like or tube-like structure. Tube-like high tensile strength. Not only give a tensile, that is high tensile strength, it also what is the tube is there through which what is the molecules, nutrient molecules diffuses from one place to other place. That is important. Diffuses other molecules from one place to other place. Also is a function like this, microtubule. Structure of mechanical strength, tensile strength. It also has a tensile strength. In another structure like this, what is that? Third one, intermediate filament is just like what is that? Looks like a rope-like structure. Rope. What is that intermediate filament it is made of? It is made of fibrous protein, not globular protein. A linear molecule. It is a linear molecule contains 310 amino acids. 310 amino acids. A linear molecule, a macromolecule, and we have 50 different protein molecules. The 50 different protein molecules means 50 different protein molecules, same amino acid, but sequence is different. Each fibrous protein behaves like a monomer here. This fibrous protein, single protein. Now undergo what is that? Association. Not one, no. Two associate like twist around like this forming a dimer. Two. Further dimer associated with tetramer, 4 like that. This behaves like a unit. A tetramer, a tetramer, a tetramer, a tetramer. These tetramers interact longitudinally. There is interaction like this. Sideways, there is longitudinal interaction. The interaction is waving, there is a weaving each other. Connection what is that? Weaving each other, but not shown this. These are connected by weaving like this. Longitudinal interaction of tetramer. So finally what is that? A thread-like structure is called protofilament. What is that? Thousands like this forming longitudinal interactions. Protofilament like this. One filament structure like this with involving all this association. After the formation of photofilament like this, here further 8 protofilaments, approximately 8 protofilaments associate laterally. How association? These protofilament wind around or twist around in a rope-like structure. All these 8 filaments, all this each filament is so much of this organization. So all 8 filaments are twist around each other in a rope-like structure, forming what is that? Intermediate filament of nanometer 10 nanometers. That is a structure we get, final structure, intermediate filament. So this is the intermediate filament is made out of fibrous protein. This filament These filaments are made up of globular protein testing matter. But all filament structures like this. These are structures, rope-like structure. They have tensile strength. These have both have elasticity. They have elastic nature as well. Elastic like there is elasticity as well as mechanical strength. Tensile strength. They have tensile strength. They can what is that used for the construction of cytoskeleton, three-dimensional structure, fish-like structure in this case. So of course these are named in different. Named in different cells as different names. This intermediate filament is named as keratin in epidermis layer, epithelial layer. Keratin is important material. They have to be because of different amino acids. So likewise, we have microfilament. It is called as actin filament in muscle cell. I told you muscle cell actin filament is important. Is involved in the conversion of that chemical energy into mechanical energy for muscular action. We got actin filament. These intermediate filaments are different names in different cells. Very important because they are the one which can withstand the cellular structure of pressure. They are called keratin in epithelial epidermis. They are called neurofilament in neuronal cells. And some other what is that in muscle cells. They are also called as what is that. They have worked as flexibility. They have what is that mechanical strength as well as flexibility. So all this together required for the construction of what is that cytoskeleton, three-dimensional jelly-like structure is important for the biological function, normal biological function of the entire cell like this. Thank you very much. Continue in next class.